those cases this morning, but we do have one matter that we want to
Okay.
address with the judge.
Please rise.
You see. There's a jail glitch. So it'll be over shortly.
Hey, everybody, cell phones are off.
Yeah, go ahead.
What time do you think you're going to be on this fall? Is that going to be Saturday?
Yeah. But no, she's coming at 11 or 11:15.
Okay, at some point, we have to go in front of the judge to get clarification as to the extent she testified. My notes are somewhat.
We can do that. We might as well do that as long as we're doing everything else this morning.
Okay, we don't have to do it right away. I mean,
We can. I mean,
It doesn't matter. I'm sure she'll be after the morning break, but it's really their choice. We can make sure to break before we do her.
That tea up here, so it comes flying in.
I got a few drops here. Nothing major.
Well, this time I got 4 times as many first.
Well, see? Tea stains right there.
Future.
That'll be an airborne.
All right, let's go on the record.
Good morning, Your Honor. We provided some cases to the court with respect to some witnesses this afternoon, and we've agreed with the state to take that up after lunch because the state needs some time to review the cases. That's fine with us. However, we do have a couple of matters that we need to take up with the court before the jury's brought in. First of all, Your Honor, we do have some additional issues that we want to put on the record regarding the voicemail messages that were played yesterday, and I also have a motion for the court regarding the playing of that evidence. But I want to be very clear about our position. In light of how the messages came out yesterday, we have dire concerns about the prejudicial effect of the messages on the jury. And to recap what the evidence was, it was approximately 20 minutes of voicemail messages left on Ms. Pietz's voicemail box. They were 37 messages in total, and it was message after message of concerned friend and family member, wondering where she was, hoping she was okay, begging and pleading for her to call, and expressing significant concern for her well-being. The content and the tone of the messages was striking, but perhaps more important is that a number of the folks who left those messages were in the courtroom, and there was a significant amount of emotional output. There was a number of people were observed crying during the playing of the messages, and I think the effect on the jury was significant. The jury sat here for 20 minutes listening to these voicemail messages. There was a screen on the TV, but there was nothing on it for them to watch, and so they had nothing to do except listen to these incredibly emotional voicemail messages worrying about her well-being. And a number of the jurors I observed looked over and observed the friends and family members who were crying during the playing of those messages. Your Honor, our position as we articulated it yesterday is that the messages are irrelevant. They're not probative of a material fact in this case. They can only be used to generate sympathy in this case. Additionally, the messages themselves are cumulative in nature. There were 37 of them. There was not a need to play all of those messages in their entirety. Given all of these factors, the content and tone of the messages, the observed impact on the jurors in light of the conditions of the courtroom, the prejudicial effect was palpable and undeniable, and this is exactly the type of matter that's to be weighed under ER 403. Even were there any minimal probative value, the prejudicial effect is substantial, and we believe that this evidence should have been excluded. We are now asking the court to grant a mistrial in light of the evidence that was played yesterday. We don't think that it can be cured, and we're asking the court to grant a mistrial.
Is that it?
That's it, Your Honor.
Okay. The motion for mistrial is denied. I explained yesterday the rationale for letting the messages in. One thing I didn't mention when I indicated that the timing of her death has been an issue in the case. I think in response to that, Mr. Offenbecker, you said that you weren't really contesting the timing of death, or you weren't taking a position on the timing of death, I think. But that is at odds with really the cross-examination of the tracker, who was called by the state to try to set a time frame for when Nicole died or when she was placed there. And there was aggressive cross-examination of her to undermine her position that the body had been there for a number of days. I think it's the implication is the defense is I won't say directly contradicting the time of death, but certainly taking a position that she died much later than is being suggested by the state. Accordingly, I think the time of death, not the precise time of day of death, but what day she died on is a key factor in this case. And as I explained, the continuing voicemails day after day to her would suggest that she was not alive to respond to those voicemails. And I agree with you, the voicemails were probing, they were emotional, but that's the reason, in my judgment, a person receiving those voicemails would reply to them if received. So I think it's highly probative on the issue of not the precise time of death, but the day of death. As to the reactions of the family, this is an open court process. I cannot prevent the family of a deceased from attending trial, and I can't control the emotions of the family of the deceased as they listen to testimony. If they are acting inappropriately, or if they are somehow acting in a way that's designed to influence the jury, then I can ask them to leave. But short of that, I cannot turn what is a death case and what is family emotions into a blank slate with no emotions in the courtroom. I can't and I won't. They are entitled to be there, and I can't tell the parents that they must absolutely blank out all emotions in response to the testimony. I think it would be an impossible thing for them to do, and the only remedy would be to exclude them from the courtroom. Motion's denied.
You know, the only other thing I want to make clear is that
Counsel, I thought I asked you if you'd said everything.
you had.
I had, and you said you had, and now you, after I've made my ruling, you want to say some more?
I just want to put on the record that Detective Decker, the tracker, said that she believed the body had been there about 7 days, and these voicemails went further into the week than that.
true. But she thought, she didn't say how long the body, she said plus or minus days. But the issue was, did she die that weekend or not? Did she die on Saturday? Did she die Friday? Did she die Sunday? And I think the cross-examination of the tracker was, to me, it implied the assertion that the tracker was wrong, that the time of death could have been far later than what the tracker was saying, and that the tracker's testimony should not be credited with establishing the time of death Friday, Saturday, or Sunday, which would have been when the tracker would have put the time of death. These emails went, or the messages went on into the week, and I think they tend to credit the argument that she was not alive to respond to those voicemails. And given the emotional impact of those voicemails, anyone who received them would likely have responded to them.
Your Honor, there's another matter, and we need to take it
Yes,
up now,
sir.
or before the witness takes the stand. This is one of those 404B issues, and I know the court, this is regarding prospective witness Michelle Beltz, B-A-L-T-Z.
Give me just a second, would you?
Yes, and this was on page 25 of the state's.
Well, that's what I'm looking for, because I have it right here. All right, I have the page of the brief. I'm looking at the proffered testimony.
And I know the court made a ruling on that. I've crossed out part of what's on the state's brief, but for the life of me, I don't have good notes on this. As far as the part where the state says that the defendant made comments about her and other female employees' bodies and compared them to his wife, such as, "I wish Nikki had boobs like that," that part I couldn't find at all in the state's proffer. I'm sorry, in the statement, tape-recorded statement made by Ms. Beltz. I have that crossed out, so I think that is that was excluded. There's also a part in her statement that she witnessed, meaning she was nearby in the gym on several occasions, one especially where David Pietz was very angry at Nicole, and he threw something against the wall, not at Nicole.
That's not included in the...
It's not included in that
trial.
proffer, but it was mentioned somewhere else in the brief, I know. And I don't know where we're at on that. To put timing on this, we're talking at least 2 years prior to Ms. Pietz's death, according to Ms. Beltz's tape-recorded police interview.
My understanding is both those things you just discussed were excluded. So I'm
What
not,
are you calling Ms. Beltz for?
because the court may
You
recall.
say Beltz, is it Baltz?
I think it's Baltz.
Okay, I got B-A-L-T-Z.
Your Honors, the court may
Right.
recall Ms. Baltz also is able to testify about comments the defendant made directly to Nikki about not doing her hair right, not running, not working out, comments he made denigrating Nikki. That's what we're calling her about, not the stuff that's been excluded. We've specifically instructed her she can't talk about the fight, argument, the throwing. We've specifically instructed her she can't talk about comments that were made like, boy, I sure wish Nikki had boobs like that. We've specifically instructed her no on that. And we've also specifically instructed her she can't talk about any sexual stuff he did towards other women at the Bally's gym. So I don't believe we have any disagreement on what is or isn't admissible.
I would tend to suggest all of it has some probative value. It all goes to the issue, but I have to make the decisions under 403 in a balancing test, and I'm doing my best to do that. And so I'm excluding some things and allowing some things in on balance.
Your Honor, I don't know if this part was raised to the court before.
Which part?
The part that Ms. Karlstrom just indicated that's what she was going to bring out.
You mean about her commenting
About-
directly to his wife about her inadequacies?
I-
Alleged inadequacies?
Yeah,
No, we discussed that. I remember discussing that at pretrial.
and I would just question, I'll address it to the court, but I would assume that these are statements that Ms. Baltz said she overheard. Is that what the state's saying, or are these statements from Ms. Pietz repeating these?
I'm understanding that she actually heard But these are statements that the witness heard the defendant say to his wife.
That's correct. I've instructed her she can't repeat things Nikki told her. And obviously, if that slips out, there'll be an objection, but I have instructed her specifically that is not to be offered.
Your Honor, then I would ask that that be, or at least reconsidered and excluded. It just seems to me something 2 years before, especially given the raft of evidence that's come in already.
This conduct occurred, and even prior to marriage. But your argument has been, on one hand, the information was too old, and then when they bring in newer information, just a few days or weeks before the death, you say, well, now it's repetitive. But it's a continuing course of behavior that apparently was almost without any long periods of interruption. That's, I think, the point they're making, is that there was never a period during the marriage where he was acting appropriately with regard to his wife and other women. And that's what this testimony is on, is that the conduct continued.
Well, Your Honor, just because the court hasn't heard any appropriate...
I said periods. I'm sure. I'm not saying everything he did in the marriage was wrong. I would never say that. But it appears to me from the testimony, from the time when they were engaged all the way up until the time shortly before the death, this type of conduct continued to occur.
Okay,
And
well,
that's the point. You're saying, well, some of the conduct is too old and some is repetitive, and I understand the argument. But the notion is that the conduct was apparently unabated.
okay, and I understand the court's ruled. I don't intend to try anymore to get the court to move off it. I would just state for the record, though, that if this is misconduct, which I don't think it is, it's in a different classification than dating other women. Husband and wives have these conversations both ways. Wives telling husbands, hey, shape up, you got a pot. That's, to me, hardly relevant if the husband dies afterwards by some reason that's suspicious.
In isolation, I would agree with you, counsel.
Thank you, Your Honor.
Record is the court. I just want to say there has not thus far been any testimony about comments he's made denigrating Nicole to Nicole, and that is not 404B. That is statements to his wife denigrating his wife. So I don't think it's cumulative and has not even come up.
Well, the question is whether it's probative, whether it's relevant. And then the issue is whether, if it's relevant, it is excluded under 403. Statements that he made are non-hearsay because they're non-hearsay under 801. So it passes the hearsay test. After passing the hearsay test, you get the relevance in 403. I'm using all those rules on balance to say that this is admissible to show not only that he had a dissatisfaction and apparently displayed it with approaching other women, but also displayed it in direct comments to his wife. And the issue ultimately is whether on January 27th or 28th of 2006, there was something that precipitated the behavior that led to the death of the wife. And that's the only reason all this is being admitted, is what was the motive either to cause a controversy or to cause a reason to want to remove his wife from his life. Anything else?
No, Your Honor.
Bring the jury in. Who will be your first witness then?
Captain Anderson.
Please come forward. Raise your right hand. Wait a minute, I think you're already under oath, aren't you? This is not the first time you've been here.
No, we're recalling, Your Honor, I'm sorry.
You're recalling him.
Calling Captain Jesse Anderson.
All right. Please be seated. Members of the jury, once a witness is under oath in a case, they remain under oath in a case until they are excused.
Just state your name again to remind the jurors.
Jesse Anderson.
And Captain Anderson, when you were previously here, you discussed some parts of the investigation into the death of Nicole Pietz. I want to ask you some additional questions about that. As part of your investigation, did you interview a witness, Christina Knudsen?
I did.
And do you recall what day you first talked to Ms. Knudsen?
It was February 13th at 5 p.m.
And what year was that?
2006.
And what was your understanding of who Ms. Knudsen was?
She worked the front desk at 24-Hour Fitness.
And when you talked to her on the, do you have your envelope in front of you? Is that what you're looking at?
I do.
When you talked to her on the 13th, was that by telephone or in person?
By phone.
And I don't want you to tell me right now what she said, but did you ask her some questions about a phone call?
February 13th, 2006.
I don't want you to tell me what she said right now, but I just want to ask, did you ask her some questions about the phone call that had been placed by Nicole Pietz's cell phone on January 28th of 2006?
I did.
And did she provide some answers to those questions?
She did.
Now, did you write down or take notes as to what she told you?
I did take notes.
And did you transfer those to the report you have in front of you now?
I did.
I want you to look at the entry that you have there for February 13th, Captain Anderson, and I would like you to read verbatim, word for word, what's written as the third sentence in that entry.
Your Honor, before he reads it, this matter's been taken up before the court already. Okay, thank you, Your Honor.
It has been, and
Ruled.
you have a continuing objection.
Thank you, Your Honor.
Go ahead and just read that third sentence if you would, Captain.
Christina said she doesn't remember Nicole calling Martin at work on 128.
Thank you, Captain. There is another question I want to ask you that I neglected to ask you last time when you testified about your contacts with the defendant at his home on February 6th of 2006. I believe in your previous testimony you talked about him giving consent for you to search his home and car on that date. Is that right?
That's correct.
And was he present? Did he stay present during that time period?
Yes, he did.
At some point during that time period, was he asked by you or Detective Decker what Nicole might have been wearing when she went missing?
Yes, Detective Decker asked that question.
And were you present when she asked that?
I was.
And what was his response?
He paused for a moment, and as a couple seconds, basically a couple seconds, as if he was in thought, and then he asked, are we implying that she was, or he asked us if we are implying that she was naked.
Okay, those were his words.
Yes.
Are you implying she's naked?
Yeah,
Had you told him prior to that her body had been found nude?
I had not.
Thank you, Captain Anderson. I don't have any other questions for you.
Yes, thank you. Captain Anderson, we met before.
Yes.
Thank you. Now, that's not all Martin said, though, right, about clothing? I mean, he said more than that, didn't he?
A little more.
Yeah, for the record.
Why don't you give us a chance to look at it?
Sure, and I
I'm
just...
trying to look at the notes to answer your question.
Well, let me just refer you to where I think you should look. Your entry on February 6, 2006 at 22:15 hours. Would you take a look at that?
I'm looking at that now.
Okay, and that's your follow-up report, I take it?
That's correct.
Okay, so let's put this all in context. Let's go back to the entry that's on February 6, 2006 at 20:28 hours, or 8:28. And I believe that's your page 20, looking at the upper right, though yours might be paginated differently. Tell me when you...
I'm sorry, what time are you referring to?
Well, on the copy of your report that I have, it says 28 hours.
Counsel, you can step forward if you want to show on the page. Sometimes these, they're after photocopying, the students get different captions.
Okay, I got it.
Okay. Context. February 6, 2006 was the day that Nicole's body was found,
That's correct.
and that was found down in Burien.
Correct.
And there were news reports that were on both the electronic news and also the print news soon after the body was found.
That I'm not familiar with. It wasn't that soon after the body was found.
Well, let's go down to your entry at 28 hours. I'm going to paraphrase it first. You arrived at the condo, yes?
Correct.
Go back a step. What time was the body first located? You can go back to your page 18. Would that be helpful?
Well, I was notified by my sergeant, Mark Toner, that the body was found at 1:13. I was notified at 1:13 p.m. that the body had been found, so I'm not, I don't have the exact time except for, I have 13:02 that's reported as the incident time, but I'm not exactly certain without looking at the CAD report whether or
Okay.
not that's the exact time.
Okay. In the CAD report, that would reflect communications between dispatch and different vehicles, different
That's
police
correct.
vehicles.
Correct.
Yes?
That's correct.
Okay. So we're talking about 1:13 when you were first notified. Is that right?
Correct.
And so this is more than 7 hours later that you went out to David Pietz's residence, yes?
That's correct.
And David Pietz told you that he heard on the news that a body had been found in Burien.
Okay, that's correct.
That's in your notes, right?
That's correct.
And he wanted to know if that was Nicole.
That's correct.
And there was nothing in your mind suspicious about him knowing it ahead of time, ahead of your being there, because the news had been out.
Yes, but it had not identified her as
Right.
Nicole.
I understand. But he said he wanted to know if it was Nicole.
That's correct.
Okay. And that's certainly an inference that one could draw if somebody's missing and there's word over the news that a body's been found and detectives are coming out to your place. I mean, it makes sense that somebody might ask, is that my wife? Is that Nicole?
Talking two different counties, it's not completely uncommon to find human remains. So
Of course.
I don't know what was going on in his mind at the time, what his thinking was, because
I'm not asking you to do so, but you're a trained investigator, and this would be something you'd be suspicious about if at the time it struck you as something that David Pietz shouldn't know anything about a body being found or connecting it with his wife.
it's not surprising that he had watched the news and had seen the footage of the body being found.
Okay. And so there was news out that day before you got out there about the body being found.
Well, there was news reporters at the scene. I wasn't watching the news, so I can only assume that there was a broadcast being made
Okay.
while I was processing the scene. So I don't know for certain, though.
And in terms of David connecting the body location with his wife, you're aware in this case that there was some information you received that she was supposed to, Nicole was supposed to go to an AA meeting down in the general area in South King County within a reasonable radius of where the body was found.
That's correct.
Now, in terms of what David said, and I'd refer you now to 22:15 hours on February 6, 2006. Can you go to that page in your report, the one you were at just previously? I'll show you on my report. It's page 22 up here.
I've mixed up my pages here when I had that on top, so.
Okay, we got that?
We got it.
We're on the same page as they said, right?
That's correct.
Okay, so you put down here, you call it a note, but it's in your report, obviously.
Right.
And so before Martin David Pietz said anything about what you're implying, there was a question by Detective Decker to Mr. Pietz, right?
That's correct.
And according to your report, Decker asked Martin about what Nicole may have been wearing when she went missing, and he said he doesn't know.
That's correct.
Is that right? And then you say he paused for a couple seconds as if in thought, and then asked us if we were implying that she was found naked.
That's correct.
In fact, the news reports indicated the body of a naked person was found in Burien, didn't they? Do you know?
I don't recall, but I'm assuming that they did.
Okay. Now, going back to your interview of Ms. Knutson, and I believe that is in your report. It's on Monday, February 13th, 2006. Do you see that?
Okay.
That would be over 2 weeks after the phone call was made on January 28th, 2006, from Ms. Pietz's BlackBerry phone to the 24-hour fitness. We're talking more than 2 weeks.
That's correct.
I'm sorry.
I missed the date that you were referencing.
Okay, you want me to start over? I'm not quite sure.
No,
Were you talking about?
February 6th being 2 weeks after the 28th? I wasn't
No,
sure
I'm
what date you
saying,
were.
okay, I can start over. The entry on your report with regard to the conversation with Ms. Pietz is on Monday, February 13th, 2006, correct?
Correct.
I'm sorry, Ms. Knutson, yes?
Yes.
And that's over 2 weeks after the time that the phone call was made on Ms. Pietz's BlackBerry, which was made on January 28th, 2006.
Okay.
That's correct?
That's correct.
And where are your notes with regard to that phone call?
I don't know if I have notes on that. It wasn't my follow-up piece on this. I think it was maybe Detective Decker who followed up on that. I don't recall.
Okay, but was it you that made the phone call to Ms. Knutson, or was it Detective Decker?
Oh, I made the phone call. I
Okay.
thought you were talking about Nicole Pietz's phone records.
No, I'm talking about your notes about the conversation with Ms. Knutson.
Yeah, correct. These are accurate notes here.
Well, but these aren't the notes you were taking when you were talking to her. These are typed.
These are typed, yes, but they're transferred over. Okay, I understand where you're coming from now. We don't save the handwritten notes. We dispose of those.
Okay, you don't have the handwritten notes
No,
anymore?
I do not.
Okay. Thank you. No further questions.
Anything else from the state?
Captain Anderson, what is the, why don't you keep the handwritten notes once you've transferred those to your follow-up?
Handwritten notes are usually pretty cryptic. They're just bullet points of information, and then we transfer those over soon after an interview to our follow-up report. So it's actually has more details in it than actually our notes do.
And do you have any doubt that you accurately put into your follow-up what Ms. Knutson said to you?
No, I don't.
Objection, meeting.
I'm sorry, what was your answer?
No, I don't.
And
Court rule?
He overruled
She
it.
said you have doubts.
Okay.
That you say I do or I don't.
counsel asked you a question and said, well, there were news reports, and they said there was a nude body. Do you recall him asking you that?
Yes.
You looked a little hesitant. Do you know for certain?
Your Honor, I would object to the editorialization provided by the prosecutor. I think that's an improper question.
Well, look a little hesitant. That's
Well,
hard.
yeah.
Yeah, well, that is improper. The jury can evaluate the demeanor of the witness. That's the objection, it's the same.
And ask that that be excluded, or at least the jury
The
disregard it.
jury is in the role to determine the demeanor of this witness. It's not the role of either lawyers to tell you what the demeanor of the witness is, your function, but you can certainly ask the question about his response without your own characterization of what you think was manifested.
Thank you, Your Honor. Are you certain that the news reports talked about whether the body was nude?
No, I'm not.
And let me ask you, typically in a homicide case, would the sheriff's office release details about a body before beginning the investigation?
No, I'm not.
Thank you, Captain. I don't have any other questions for you.
Sheriff, it's, or Detective, it's not uncommon, though, for the news media out at the scene to have information that the official sheriff's department has not disclosed to the media, isn't it?
They may receive information from witnesses or just their own observations and report out on those. That's possible, yes.
And in this case, there was a civilian witness who found the body, wasn't there?
There was. It was transported away from the scene.
Okay. And the media also can listen to police dispatch reports through use of scanners, can't they?
Yes, but this information would not be released over
Well, but-
police radio.
First report from the sheriff out at the scene could be to the dispatch, we found a body, we found a nude body.
Yeah, I don't have the CAD report in front of me to tell you whether or not that was relayed by the witness to our communications center.
And it's not uncommon also for the media to have anonymous sources in the sheriff's or police departments. That happens a lot, doesn't it?
Not so much with our agency, but it does happen.
Okay. And you said there was media around.
Yes.
Was there a news helicopter also?
I don't recall for certain, probably.
Okay.
But it's right close to the flight path there for SeaTac Airport, so they can only get so close in that area.
I see. Okay, thank you. No further questions.
You may step down.
Thank you.
Please don't excuse the witness because we may have to call him in our case.
You might be called back a third time.
All right.
Or fourth time. You're not excused.
Thank you.
Did you have any papers there, Detective?
These are my own requests.
Okay, all right. Come forward. Your testimony in this matter will be the truth, the whole truth, and nothing but the truth.
I do swear.
Be seated.
Good morning.
Good morning.
Can you state your first and last name and spell your last for us?
James Curry, C-U-R-R-I-E.
And Mr. Curry, what do you currently do for a living?
I am a forensic biologist with the Center of Forensic Sciences in Toronto, Canada.
How long have you been in that job?
With that particular employer, I've been there for just over 3 years.
Did you at some point work for the Washington State Patrol Crime Lab down in this area?
Yes, I did.
What time period did you do that?
Between 2002 and 2010.
What was your job, or what were your job duties when you were with the Washington Crime Lab?
When I was with the crime lab in Washington, my duties were also a forensic biologist and DNA analyst.
I'm sorry, what?
Forensic biologist and DNA analyst.
And what does that mean in terms of day-to-day duties? What did you do when you were with the lab here?
My primary duties were to identify body fluids, or attempt to identify body fluids, and to assist in establishing who might be the source of that body fluid using DNA technology.
Is that the same thing you do now up in Canada?
It is.
Do you do forensic work and look at evidence from crime scenes up there as well?
Yes, I do.
Can you tell me a little bit about your educational background that prepared you for this DNA work?
Yes, I have a Bachelor of Science degree from the University of British Columbia in Vancouver, Canada, in the area of pharmacology and therapeutics. And that sounds like an odd degree to have for a forensic biologist. It's a research degree that focuses on drug development and drug design. And to effectively design a drug, you need to understand how the human body works. And some of the most potent drugs that we use today are anti-cancer drugs and antiviral drugs that target specifically DNA. So you need to understand how DNA works in order to design a drug. So with that, I have a very rich background in molecular biology, biochemistry, and biology and genetics. And that's how I came to the field of forensics.
And was the Washington State Patrol Crime Lab your first job where you were doing DNA work, or did you have jobs before that that involved that?
That was my first job doing DNA work, yes.
And did you receive training through that lab as to how to do your position and how to analyze things for DNA?
Yes, part of being employed by any accredited forensic laboratory is that you have a certain educational background. You're also required to undergo a one-year training program that teaches you the principles of body fluid identification and forensic DNA analysis.
Did you, well, obviously you were at the lab in 2006. Do you recall what your title was at the crime lab in 2006?
It would have been a forensic biologist or forensic scientist.
And did you examine items related to the Nicole Pietz homicide case?
Yes, I did.
I want to ask you a few general questions first about DNA. You talked about bodily fluids. What types of things from our body can be looked at to glean a DNA profile?
I'll just step a little bit and just give a little overview of DNA, just so we're all on the same page. DNA, I'm sure most of you know, is found within each one of us. And because it has information about our growth and our development, it's often called the blueprint of life. Within the human body, DNA is found within virtually every cell. So using myself as an example, I have DNA in my blood, I have DNA in my saliva, I have DNA on my hair roots, and DNA in my skin, and so on, every single cell type virtually. Within the human body, DNA is the same regardless of the location. So again, using myself as an example, the DNA in my blood is the same as the DNA in my saliva, which is also the same as the DNA on my skin or in my skin. And that's an important concept because I know that if I examine a sample of blood and a sample of saliva and they originate from the same person, then DNA profiles will match. DNA can be found in blood, as I said, saliva, and semen. And those are the primary body fluids that we examine in a forensic laboratory. It can also be found in occasions when you touch something or you handle something. We generally call that handler DNA, or if it's found in a garment, we call that wearer DNA. It's left behind by skin cells. Now, the type of DNA that I've examined for this particular case is found within the central portion of a cell, and we inherit this type of DNA in equal parts from both of our parents. So 50% from your mother, 50% from your father. It's not predetermined which portions of your parents' DNA you will inherit. That process is entirely random. And that's what guarantees that the DNA within you is unique, this random inheritance. The single exception being identical twins. Now, having said that the DNA within us is unique, in fact, 99% of DNA within us is not unique. We have that much in common with each other. It's that remaining 1% that is very variable and hypervariable and is different from person to person. And it's that 1% that is the target of my DNA analysis or any forensic laboratory DNA analysis when they undergo their testing, because this is what is different from person to person. The primary goal of my analysis is to determine a DNA profile from evidence. And having done that, if I have multiple DNA profiles, do they match or do they not match? If two DNA profiles do not match, the conclusion is they come from different people. And that is absolute. There is no statistic. If two DNA profiles do match, there are two explanations. They match because they come from the same person, or they match because the DNA profiles come from different people and they match by coincidence. My goal as a forensic DNA analyst is to give to you the significance of any matching DNA profiles. How significant or how likely is that coincidental match? And that's expressed as a random match probability, and that's essentially how rare is a DNA profile in the general population. All that information is generally contained within a forensic DNA report, which is
That's a lot of information, but I want to go over a couple of things. The first is you talked about the fact that 99% of our DNA is not dissimilar from others, but 1% is. The work that you do you focus on the locations that will give you the unique profile then, the 1%?
yes, that's the target of our examination, yes.
And when you said that if two profiles match, it's either because it's the same person, or you said it's a coincidence, and by coincidence, are you able to apply numbers to the likelihood of finding that same combination or profile in the population?
Yes.
Okay, so is that done to avoid knowing whether, or to find out whether it's a match versus a coincidence?
It just simply tells you, essentially it tells you how significant is the match. Is it a coincidental match, or is there strong scientific support that the person in question is the source of the DNA profile? And that's expressed as how rare is the DNA profile in the general population. If it's a common DNA profile, it may quite well match by coincidence. If it's a rare DNA profile, the likelihood of a DNA profile matching by coincidence is somewhat less.
And how do you tell if it's a rare or common?
It's, those are probably not the best terms. We express it in terms of a random match probability. And what I do is I generate a number. A profile may be expressed as 1 in 1 billion, 1 in 1 quadrillion. I simply present you the numbers, and it's up to the jury and the judge to determine what is the significance, what is the weight that they will give that number. I can tell you that the more smaller the number, remember we're talking about 1 over a large number, so the more smaller a number, the more, or the stronger the scientific support that a person is the source of a DNA profile, and it's not a coincidental match.
So in other words, if you say there's a 1 in billion chance that there's someone else that matches this besides this person, the likelihood is it's them because of that billion is such a large number. Is that fair to say?
The more precise way is just to express it as the frequency of the expected frequency of the DNA profile in the population.
So let me ask you this. You talked about different sources that you might examine for DNA. You talked about blood, semen, skin cells, I guess, when you touch something. Do you follow the same process to obtain a profile from those different various areas?
Yes, generally the process is the same in terms of DNA analysis and extraction. The single exception being for samples that originate or might potentially contain spermatozoa or semen. There's a little difference in how DNA is dealt with in that one instance.
So can you tell us then generally the process you use, and as basically as you can, and let's stay away from the extra thing you do for the spermatozoa. What do you do? What's the first thing you do when you receive an item and you're attempting to find out if there is DNA, and then you're attempting to get some type of profile if you can?
Correct. All right. The first thing you do when you receive an evidence item is you examine it to determine if body fluid is present. And that can be done through visual observation as your first cue, and that's generally followed up with either a chemical test of some sort. So we have chemical tests to assist in identifying blood, chemical tests to assist in identifying saliva. Having done that, the next thing you need to do is you need to remove the sample. When you have this sample containing your possible blood, your body fluid, you need to extract the DNA from the cells in that sample, which we do chemically. Having done that, you need to determine how much DNA is in that sample. Too little, your results will be difficult to interpret, possibly unpredictable. Too much, there's a potential to overwhelm the instrumentation that you use further down the line. Having established how much DNA is in the sample, then, as I mentioned, we target the variable regions of our DNA. What we do is we amplify specific sequences of DNA within that variable region. Sometimes we amplify 9 locations, sometimes we amplify 13 locations. And when I say amplify, it's essentially a photocopy method where you make millions and millions of copies, or millions of copies of your area of interest. We call that polymerase chain reaction, or PCR. And the areas that we examine are called short tandem repeats, or STR. And the locations that we examine, we generally call loci, L-O-C-I. And it's just another word for a test location. Having done that, we need to separate out the DNA fragments that we've amplified to determine if they originate from a single source or one person, or if they're a mixture or they originate from multiple people. And then I produce results in my report.
So the process of copying that I believe you called that the PCR process?
Yes.
Does that allow you to take what might be a very, very small amount of DNA that ordinarily you might not be able to get a profile from, but by copying it over and over, you can, or more likely to be able to obtain a profile?
Correct. It assists in those cases where there's a small amount of DNA present on an item. Using this process, by making millions of copies, you make it easier to analyze.
And I want to talk about the work you did in this case, but I want to ask a couple more questions just generally, and that is, you talked about touching something and possibly leaving DNA. Do you always leave your DNA if you touch something?
No.
Are there factors that can affect whether, for example, me touching right here is going to leave my DNA on that surface?
There are a number of variables involved, and I should say you don't always leave detectable amounts of DNA. So methods that I can detect in my laboratory. So there are a number of factors involved. We each shed or leave behind different numbers of cells when we touch something. Some people readily shed cells, some people don't. Is there moisture? Are your fingers sweaty? Because while the sweat doesn't contain DNA, it does act as it assists in moving DNA or cells from your fingers to a surface. What's the length of time that you're in contact with the surface? How rough or rigid is the surface? The more rough, the more likely you're to coax cells off. The smoother surface, not so likely. So those are generally some of the factors we consider in terms of what we call handler or touch DNA.
Does temperature affect it at all? Either temperature of you kind of talked about temperature of the person or temperature of the surface. Does that affect at all the likelihood of leaving it?
Not really, only in the sense that if it's a hot surface, you might go, ouch, and then sort of brief contact.
And can DNA, assuming you leave something or leave a detectable amount, can it go away over time? Does that happen?
Yes, it can. DNA is surprisingly stable. We've detected DNA from pharaohs, from mummies. So DNA by itself is very stable. However, it is very prone to attack by moisture, by bacteria, by fungus, by sunlight. All of these factors can cause DNA to degrade over time. And that's the expectation when something's left outdoors or in a warm environment, that over time DNA will degrade.
Is there any known or set formula that you can say, oh, after this period of time, under these conditions, there'll no longer be detectable DNA?
No.
Can a person leave a fingerprint but still not leave a detectable amount of DNA?
Yes.
If somebody touches something and then another person subsequently touches in that same area, could the second person rub off or destroy a detectable amount of DNA from the first person?
There are two possibilities. If I were to touch a surface and leave behind DNA, and somebody were to come after me and touch where I've touched, they could either overwhelm or mask my DNA, so leave more DNA behind than what I left behind, so that when it's analyzed in the DNA lab, the second DNA profile may be in much higher quantity than the first DNA profile, and it may not detect the first DNA profile. The other possibility is yes, depending on and generally expect in moist conditions, like if there's moisture so that it's transferred to the second person, they can potentially remove a portion of that first DNA deposition.
When you examine items, and let's focus on this case, Did you receive a lot of items to examine that came in a form called swabs?
Yes.
Can you just explain what a swab is?
A swab, very basically, it resembles a Q-tip swab. Obviously, they're not Q-tip swabs, but that's what they look like. And they're sterile. There's no DNA on them, or there's not supposed to be any DNA on them, and they're just simply used primarily for internal samples or to collect small samples. And when I say internal samples, oral samples, vaginal samples, or anal samples.
And is that something that you will receive sometimes when it's been swabbed by, for example, law enforcement
Yes,
or the medical examiner, perhaps?
correct.
Do you yourself sometimes actually have to also examine items yourself, and then do you or your office also use swabs?
Yes.
When you are examining swabs and attempting to get a profile, I guess what do you do? And you talked about the general process, but with a swab, what do you do to try to pull off any potential DNA that might be on that swab?
The process is, when we receive the swab, it is dry because we don't want any DNA degradation to occur from the time of collection to the time I've analyzed it. So it's dry. What we do is we immerse it in some kind of buffer or solution, and that coaxes the cells off the swab. We want to get as much material off the swab that we can so that I can perform my analysis downstream.
When you receive swabs or actual evidence items from a crime scene, are you always able to get a DNA profile?
No.
Can it range from getting a full profile to getting what might be called a partial profile?
Yes.
Or sometimes is it possible to not get enough DNA to detect and get any kind of profile?
Correct.
Just so we're clear, what would a partial profile mean? What would that mean?
A partial profile is essentially not a full DNA profile. And when I mentioned that we test the variable regions of DNA, for most of this case, I've tested 9 specific locations in that variable region. So a full DNA profile will be results of all 9 locations. A partial is I may have received results of 7 and not the remaining 2.
And is there, you talked about sort of detectable levels. When you are trying to talk about describing whether you get information at all 9 locations or only certain locations, is there a certain level by which you have to say it has to be this much for me to count it at this level? Does that make any sense, my question?
No, maybe if you
Is there
phrase-
like a certain peak that has to be reached over a certain level before you're able to say, this is reliable and I can consider it information at this level?
Yes, excuse me. Yes, as part of the process of analyzing the amplified DNA, there needs to be a certain amount of DNA, or it needs to cross a certain threshold before I can reliably conclude that DNA is present at that test location.
So are there situations where you might have some DNA detected at every location and you might be able to tell whether it's consistent with another profile, but it might not be high enough to count officially in your ultimate result? Does that make sense?
Yes. Obviously, when we analyze the DNA profile, based on laboratory tests, we determine what the threshold is, at what point we're confident that you have DNA present. Now, that doesn't mean that what falls below the threshold isn't DNA. We know that obviously it will be DNA. But there's also the possibility that it could be artifactual or just part of the amplification process. And so I can still see the results, and I can still determine that's possibly the same as individual A or individual B, but because it doesn't cross the threshold, it's not reportable as a DNA profile.
What is a reference sample?
A reference sample is a known sample collected from a person, primarily used for comparison to unknown samples.
And why might you receive reference samples in a criminal case?
It's to determine, can you match the unknown profiles to persons of interest, to either an accused, to a complainant, to a consensual partner, to any person of interest, to try to associate, to see if any associations can be made to DNA profiles.
And so when you get the reference sample, do you find out what that profile is, so then you can compare it to anything you do on evidence items?
That is correct.
I know you don't work for the Washington State Patrol Crime Lab any longer, but are there any resource issues that affect how many items law enforcement might be able to submit to you that you can realistically examine?
Yes. Essentially, as a scientist, it's my job to determine, and this falls true for the Washington State Patrol Crime Laboratory, it falls to the scientists to determine what is the question being posed, and which evidence items will best assist in answering that question. And so it is up to the frontline DNA scientist or biologist to say, those evidence items may not answer that question, they're not necessarily probative. These are the ones that may assist in answering our question. And so those will be the ones that are tested. It's an art of negotiation. Obviously, law enforcement agencies would like everything tested, but crime laboratories don't have the resources to throw everything at one case that involves 1,000 items and then focus on another case that involves 1,000 items. So it is using what I know as a scientist to assist in answering the question posed by the most probative pieces of evidence.
And in the case involving the materials submitted in the Nicole Pietz homicide investigation, were you what you just described as sort of the frontline or main DNA scientist who was going to look at this case?
Correct, yes.
And in this case, did you receive some reference samples to analyze?
Yes, I did.
And can you say what those were that you received?
Yes, I received one reference sample from Nicole Pietz and one reference sample from David Pietz.
For Nicole Pietz, what form did that reference sample come in?
May I consult my notes?
You certainly can.
The sample from Nicole Pietz was a dried stain received on a card, a dried blood stain, sorry.
Did that come from the medical examiner's office, from what you could tell?
Based on the item designation ME, that's my understanding, yes.
And is that in a homicide case where you're dealing with a reference sample from a homicide victim, is it pretty typical to get that type of reference sample, the blood stain card?
Yes, those are generally, generally collected at autopsy, yes.
And there's water if you need it. I'm
Okay.
not sure if there's cups. Are there cups in there? And the next question, Mr. Curry, was the form that the reference sample from David Pietz came to you.
Thank you. It's described as a buccal swab. So a buccal swab is an oral swab from the mouth.
And are you familiar with how DNA is taken from an individual using a buccal swab?
Generally, it's a swabbing of the inside cheek.
And is it the same type of swab that we've talked about where attempts to get DNA are swabbed at crime scenes? Or is it a different type of swab?
It's the same type of swab, yes.
For the reference samples that you got from Nicole Pietz and David Pietz, did you then go through the process that you do and get their profiles, their DNA profiles?
I did, yes.
And as you continued to work on items, did you eventually compare the reference samples to items that you examined in this case?
Yes, I did. I should clarify, with respect to the swab from David Pietz, it's described as a buccal swab, but it's actually, on photograph, it's a plastic applicator. So that's just simply another means of collecting a sample.
Would that still be from the cheek area?
Yeah, that's my understanding, yes.
Okay. Did you receive, we talked about the item of Nicole Pietz's reference sample being from the medical examiner, being labeled ME. Did you receive a variety of items labeled ME to examine in this case?
Yes, I did.
And I want to start talking about those. I'm going to show you. Mr. Curry, I want to start going through what you did in this case and the results you obtained. Can you take a look at Exhibit 97 and tell me, having this in front of the jury while you testify would help to explain to the jury and the results that you obtained?
Yes, this is a good summary of my testing results.
Peter, I would offer this, just this PowerPoint, just for illustrative purposes as he's testifying.
And they use it for electronic purposes.
Thank you, Your Honor.
Do we have an exhibit number, Your Honor?
It's a 97.
So, Mr. Curry, I want to start. Did you receive items labeled ME1, ME2, and ME3 to examine?
Yes, I did.
And can you indicate what those items were?
ME1 is described as an anterior neck swab. ME2 is described as a, excuse me, as a right neck swab. And ME3 is described as a left neck swab, all from Nicole Pietz.
And what's your understanding of what anterior would mean? I always get confused. Is that front or back of the neck area?
Anterior would be this portion, the front of the neck.
And you said swabs, so I'm assuming that came in the form of swabs?
That is correct.
Now, did you look at these all individually, or did you combine these to do work?
The initial approach was to examine each individually and then to combine them for the purpose of DNA analysis, since they originate from the same anatomical location.
And when you say that you first examined them each individually, what was the purpose of doing that, to look at them individually initially?
To determine the state of the swabs. Is there any staining on them? What is the condition of the swab?
Did you note any staining or anything unusual on the 3 swabs that you just discussed?
Yes. All swabs appear to be coated with what appear to be fibers, dirt, debris, possibly green leaf-like material, so forest-type material. And that was in all 3 swabs examined.
And did you then combine them for the purposes of attempting to extract the DNA from them?
Yes, I did.
And did you follow the process you discussed earlier regarding what you go through for that?
Yes.
And were you able to get any detectable amount of DNA from these 3 combined swabs?
A trace amount of DNA was detected from those combined swabs.
And when you say trace amount, was it enough that you were able to compare it to any of the reference samples you received?
No, I was not.
So when you say trace amount, does that mean that you were able to detect DNA even before you went through that copying process that you described?
Yes, DNA was detected on the sample. However, when I took it forward for amplification and analysis, I did not develop a full profile. In fact, it was a profile at only 1 test location.
So your actual result is, as you said, trace DNA.
Correct.
I want to talk next about an item labeled ME4 and ask if you examined that and if you could tell us what that is.
Yes. ME4 is a vaginal swab from Nicole Pietz.
And what's your understanding of what a vaginal swab means in terms of how it was obtained or where it came from?
A vaginal swab is generally collected within the vagina, and a speculum is used to avoid contamination with the exterior, so that the focus is purely the vaginal contents.
And did you examine this swab as well?
Yes, I did.
And what are you looking for when you examine a vaginal swab?
With this type of swab, they're examined to determine if semen is present.
How do you determine that from visually looking at it? What are you looking for?
When I visually look at it, obviously, it's not something I can identify visually. There are steps that we undertake, and we look for, first we examine the swab microscopically. We prepare a portion of the swab and we put it on a microscope slide to see if we can identify the cellular components of semen, that is spermatozoa, and that was done in this case. The next step, if none was detected, and none was detected, the next step is to look for the chemical components of semen. And in this instance, none of those chemical components were identified.
So let me just make sure I understand. You didn't visually see any sperm, and then you said you did a chemical test that will, first of all, is that got a particular name, the chemical test you're talking about?
Yes. One of the chemicals that we examined for is called P30, P30, or prostate-specific antigen. And that's a chemical that's found in high levels in semen. And examining for that, none was detected on the vaginal swab.
So when you examine a swab like this and don't see sperm, do you still always do the P30 test just to make sure that there's not semen there?
Yes. In cases of a vasectomy, semen is still produced, but the cellular component is either absent or a very low amount. The chemical component will always be there. So that's why we examine for both.
Does a positive P30 test, meaning that it came out positive, I know you said it didn't with the swab we're talking about, but if it does come out positive, does that always mean it's positive because of the presence of semen?
At the time of this report, if you identified P30, the conclusion would be that semen was present. I can revise that conclusion now. We now know through further studies in Canada and the United States that P30 is found in high levels in semen, but it's also found in trace levels in other body fluids and in females, specifically breast tissue, within blood serum, vaginal secretions. The difference being the magnitude or the amount of P30 present. So now if you didn't detect spermatozoa and you went on to do a P30 test and you had a positive result, it's an indication that semen may be present, recognizing that other body tissues do contain P30.
And then would you go forward to try to see if you can detect any male DNA to confirm whether or not that positive P30 was from semen or not?
At the time of this report, you would take it forward, this sample. If you identified a positive P30, yes, you would take that forward for DNA analysis. Now we have additional techniques where we can look specifically for male DNA. That wasn't the case at the time of this report.
So in other words, at this point, you can say that a positive P30 doesn't necessarily mean the presence of semen.
Correct.
So on ME4, you said there was no sperm seen and also no semen, is that correct?
Correct.
I want to ask you next about item ME5. Is that an item that you received and examined in this case?
Yes. ME5 is our anal swabs from Nicole Pietz.
And that would be swabbings from her buttock area, okay.
Correct.
Do you follow the same process with anal swabs that you just discussed for vaginal in terms of looking for sperm, looking for semen, or trying to test for semen?
Yes, I do.
Did you look visually in this, on the anal swabs to see if you could detect any sperm?
I did, and none were detected.
Did you do the P30 test on the anal swabs?
Yes, I did.
And how did that come out?
This result, the P30 came back as a positive result.
And again, as you discussed earlier, we don't know what that means at this point.
Your Honor, objection, that's leading the witness.
Counsel, it's a leading question. It was what he indicated earlier. That's a predicate, I think, to her next question.
Well, I thought she misstated his conclusion, so I believe that the leading is improper there.
She can re-ask the question. I don't think she misstated it, but she can re-ask the question.
Did I misstate what you said earlier, that does a positive P30 necessarily mean the presence of semen?
Your Honor, that was a different question that she asked this time. I don't have an objection to that.
It's okay.
You can answer that.
No, it does. A positive P30 does not mean the semen is present, absent spermatozoa.
And did you actually go forward with the anal swab sample to determine if you could get any type of DNA profile?
Yes, I did.
And were you able to obtain any type of DNA profile from the anal swab?
Yes, I did obtain DNA profile. Yes.
And what did you get from the anal swab in terms of a DNA? Well, let me ask you, did it match or consistent with any of the reference samples that you had?
It matches the DNA profile obtained from Nicole Pietz.
And was there any male DNA detected when you took this forward?
No.
So no sperm, no male DNA, but it did match Nicole Pietz?
Correct.
And I should ask, and I think I neglected to ask you on the vaginal swab, did you take the vaginal swab once you confirmed no sperm and P30 was negative? Did you go forward to attempt to get any DNA profile on the vaginal swab?
Not on the vaginal swabs.
And why is that you didn't do it on that?
The question being asked, is spermatozoa present or is semen present? I've established that semen is not present, and so my expectation is the only profile developed would be that of Nicole Pietz.
I want to go forward now and ask you about an item labeled ME6. Did you get that item and examine that item, Mr. Curry?
Yes, I did.
And what was that item?
It is described as oral swabs from Nicole Pietz.
And what is your understanding of where oral swabs come from?
Those are from the mouth.
And did you also look at the oral swabs microscopically?
Yes, I did.
And were you attempting again to look for presence of semen through seeing sperm?
Correct.
Did you observe any sperm?
I did not.
And did you do a P30 test on that swab?
Yes, I did.
And what was the result?
The result was negative.
Did you take that forward to attempt to get DNA then?
I did not.
Was that for the same reason that you just described, because of the negative P30?
Correct.
I next want to ask about items E8. Did you receive those?
Yes, I did.
What were those?
ME7 and both ME7 and ME8 are vaginal washes from Nicole Pietz.
And what does that mean?
It's simply a washing of the vaginal cavity and, a collection of fluid, the resulting fluid.
Does it come in the form of a swab or some other form to you?
It was received as a tube containing white liquid or cloudy liquid.
And did you examine that visually under a microscope as well?
Yes, I did.
Any sperm or indication of semen in that sample?
No.
Did you do a P30 test?
I did, yes.
And it was negative?
It was negative.
Did you take those samples forward to attempt to see if you could get any type of DNA profile?
Excuse me. I did, which seems a little odd given what I've said. Typical sample received at the crime lab in Washington State.
Also, is this a good time for morning break?
Yeah, it's fine, Your Honor. We can stop right now and do the break.
Thank you.